Hello, my biology students, and this is Mrs. Shaw, and we're going to start diving in to wrapping things up from last week's lecture and then moving in to this week's lecture. I've backed this up to slide 34, which I know we've already talked about, but I'm going to slide through to make sure that you see the different pieces of evidence that's coming out of chapter 14, the last of which we didn't have time to talk about on Friday. So the first piece of evidence is from the fossil record, right? And even Darwin knew this. He knew that fossils were the evidence of creatures that were living in the past, plants and animals. Obviously now we can actually find microfossils and things like that of things that are even smaller. So that's one piece of evidence that we talked about. The other we talked about a lot more on Monday about that clue of the biogeographical piece, where when we look at the distribution of organisms around the world, how it fits together, that they spread out over time, changing into those specific environments. Then we talked about a little bit on anatomical evidence right at the end of class Friday, and there's a few pieces about anatomical evidence called vestigial structures, which are those anatomical features that don't have a function, or they don't have their original function, but yet they're kind of leftovers, meaning that they're no longer necessary for survival, but they don't just disappear because we don't use them. Remember, that's a bit more Lamarckian than what we know today of natural selection. It's more the idea that other features are selected for, that are more, those traits are more useful for survival. We also talk a lot about homologous structures, and this is the idea that even when you look at them on the outside, they may look and behave very differently, but when we dive into the anatomy, we find some similarities. Now, I don't want to say that a bat wing and a bird wing is the same, but yet structurally there are some commonalities of one long, thicker bone branching into two bones and then branching into many. And we'll see that in not just bird wings and bat wings, but also whale flippers and cat legs. I know it's a lot different on a whale. You've got to look on the outside and realize that it's a bit different on a whale as well. And this is a horse leg compared to a human hand. Then we also talked about that there are significant similarities in early development, especially when we're thinking about vertebrates and tetrapods. Now, we can't be comparing this to insects. That is a very different group. That common ancestor is long ago, but we can use that as evidence that those first few milestones of development are very similar across the vertebrates, especially tetrapods, ones that have four appendages. So these are the two last slides that I didn't want to rush them on Friday. So Darwin didn't know anything about the molecular evidence of natural selection and evolution, but what we're finding out now is that all living things are kind of tied together by a few types of molecules, some of which we've already talked about, right? That the DNA, whether it's in a nucleus or not, have the same four bases, whether you're bacteria, an oak tree, a butterfly, or a human. That important molecule of ATP, which is the energy molecule. Remember, we produce that in the mitochondria, and then we use that molecule for energy of doing everything in the cell. Even cells that don't have mitochondria have ways of generating ATP. And a lot of the enzymes, which are proteins, which are made from DNA, are nearly identical. And it's interesting because we can actually sequence some of those enzymes. We can see which order of the amino acids they're in to see if there's any slight differences among individuals. When we look at the individual genes, there's a lot of variety based on the species you are. However, there are a group of genes that we call Hox genes. And these are those early genes that get expressed in order to say, hey, a head goes here, and an arm goes here, and another arm goes there, a leg here, a leg there, a tail there. So those basic body plans are dictated by Hox genes. Cytochrome C is a molecule that is necessary for the generation of ATP. We didn't talk about the electron transport chain because we skipped that chapter. But cytochrome C is an enzyme that all organisms have. We're pretty sure that all organisms have it. So this is really cool because we can take the sequencing of the amino acids of cytochrome C and make the hypothesis of, well, if two different species have very similar cytochrome C, their common ancestor was more recent than two organisms that have very different cytochrome C sequencing. And of course, we can always go back to the gene that produces cytochrome C and sequence it. The advantage of looking at the amino acid order versus the DNA order is, well, there's just fewer, right? The DNA gene can be thousands of bases long, and then it gets turned into mRNA, and then it gets edited. But then when we turn it into amino acids, remember there's 3 bases of mRNA that codes for 1 amino acid. So the protein, looking at the amino acids within that protein, it's just a shorter sequence. Now, obviously, it can glaze over some of the subtle mutations that are in the DNA, but it's a good place to start. And so here's the idea with the cytochrome C. If you compare human cytochrome C to a monkey's, and this is just real general, just types of organisms, you'll see that there are some differences, but not very many. Now, if you compare humans with pigs, which are both mammals, so we're in the same larger group, we're going to have quite a few more differences. So over here on the y-axis, it'll say number of amino acids differences compared to humans. So if we compare two humans, they're going to have the same. It's not going to show any kind of identity. Now, when we compare them to the birds, there's a few more differences. When we compare to reptiles, like a turtle, a few more. Fish, insects, and then fungus, there's a lot of differences. But yet it's kind of crazy that all of these organisms and more have cytochrome C. So it's pretty crazy. Okay, well, I'm going to switch gears. I'm going to continue recording. We're going to go into the next lecture. And let me flip over to this here. Oh, it looks like I need to... My screen's a little smaller here. Let's see if I can get it all in there. Okay. All right, so chapter 15 is a continuation of studying evolution. This chapter is called Evolution on a Small Scale, and then the next chapter is going to be Evolution on a Larger Scale. And so a lot of this is repeats that we talked about in the last lecture. So there's going to be some slides that I may go pretty quickly on because we've actually already covered it. This is really the start of Darwin's theory of natural selection, where he saw these small changes in organisms over time. Okay? Let me see if I can do this. That's probably what I need to do. Sorry for moving the screen so much. Hopefully I'm not making anybody seasick here. Okay, so let's continue. This lecture is on Moodle, and so you can download this. It's called Lecture 12. Okay, so nice little review, but this really shouldn't be anything shocking. Natural selection is this process that the end result is showing these adaptations that have built up. They may be pressured by the biotic parts of the environment or the abiotic parts. Now, these are new words. Bio means life, so a biotic part of the environment means the living part. So maybe predator-prey relationship, maybe however else they get their food, maybe disease that might be in the environment. Abiotic are the non-living parts of the environment. And of course, we think about climate and weather. If you're a plant, the soil chemistry might be important as well. Darwin's mechanism for evolution is natural selection. So again, sometimes we like to say the theory of natural selection, the theory of evolution through natural selection. It's the mechanism. It's the how it's occurring. So the idea is, over time, most fit individuals become more prevalent in a population. So again, remember that definition of fit is that they have the most babies. Because again, the direct correlation is the more offspring you produce, it's because you've gotten more resources to give your body the energy to go through reproduction. And the reason you're getting more resources is because you have some kind of advantage in that environment. And of course, this increase of more prevalent fit individuals leads to change in the population little by little over time. Because again, the most fit individuals reproduce more, and so there's going to be more of their offspring in the next generation, and they will look similar to the parents. So here's again a nice little breakdown of what natural selection involves. Number one, we have to have variation. Members of the population have to be different in some way, shape, or form, but especially in the qualities it takes to survive. The second part is we have this process of inheritance, that genetic differences in the parent has to be able to be passed down. It's not something that the parent learned or acquired or changed on its own body through its lifetime of experience, but it has to go deep into the DNA. Because if it's something that can't be passed down, well, it's not going to change the population, right? Then we have to have this pressure of increased fitness. That, again, if you are more or better adapted for the environment, you're more likely to reproduce, and those offspring will make a greater proportion of the next generation. Okay? I'm going to do this next slide, and then we'll go into the specifics in the next lecture. So, as we talked about in the genetics unit, most traits, they're polygenic. It's not one gene dictating one trait. Now, there are some exceptions, and we'll talk about them, but for the most part, it's because we have this range of variability. It's not yes, you have it, or no, you don't. So whenever we have this range of traits, those are polygenic. Those are ones that are controlled by more than one gene, more than one pair of alleles, and they're probably on different chromosomes. And so when we talked about this a little bit with height. Remember, we talked about if we took everybody and lined them up by height, we would have this bell curve, right? That we would have some that are really small and some that are really tall, but pretty much everybody else is in the middle, ranging in those average heights, and so we would have this nice bell curve. Well, in evolution, when that bell curve changes, that's showing change over time. If that bell curve shifts, if we did that same height experiment 200 years ago, probably the bell curve would be shifted towards the shorter side, right? Because over time we've changed our average height, mostly because of medical care, right, and nutrition. And so when we look at these bell-shaped curves of the phenotypes of the population, of what they look like, we see three patterns that emerge: directional, stabilizing, and disruptive. I'm going to go ahead and show the pictures, and then we'll go into more details in the next segment. So the top row shows just this nice, pretty bell curve, okay? Stabilizing is when the average becomes most common. A stabilizing selection means that the peak narrows, it kind of gets slurped in, and there's even fewer at the extremes. Do you see that the extreme little and big, and this is all general, is now being pulled up to the top. Directional means you shift to the left or the right, kind of like the height thing, right? That maybe it used to be the average was over here, and now we've shifted over time and become taller. I shouldn't say we've become taller, because that's not really how it works, right? But over time, our offspring have become taller. Disruptive selection is essentially the opposite of stabilizing. This is when parts of the extreme are more beneficial, the traits here, and what's in the middle is not beneficial. Okay, and we're going to go into more details on all of this, but I'm going to pause this video, and this will be part 1.